How can we use fluorescence imaging for early cancer detection in the digestive tract? My name is Thomas Wang. I'm a professor of internal medicine, biomedical engineering, and mechanical engineering at the University of Michigan. I have nothing to declare. So cancer in the digestive tract arises from a number of conditions that increase the risk for malignant transformation. These conditions include Barrett's esophagus, indeterminate biliary strictures, atrophic gastritis, Crohn's colitis, pancreatic cysts, sporadic colorectal cancer, and ulcerative colitis. Cancer arises from the epithelium, which is the thin lining of tissue on the inside of the hollow organs that can be accessed directly with medical endoscopes. We'll first talk about early detection of colon cancer, and a white light colonoscope is shown here, visualizing a pre-malignant lesion that consists of a polyp. This is a anatomical abnormality that is grossly visible from the endoscopic image and is removed using either a snare or electrocautery techniques. However, of increasing interest is the flat adenoma, where the epithelium does not produce polyps, but it's flat but still has molecular and genetic changes that can give rise to colorectal cancer. How do we detect these lesions? In our lab, we're developing a strategy to image the cell surface targets. These are targets that are actually proteins that can be expressed at a basal level in the normal state, but when the cell transforms into cancer, will be highly overexpressed and thus be available for imaging. There are a number of ligand platforms that can be used to target these proteins, including antibodies, aptamers, lectins, and peptides. We are developing peptides as imaging agents and labeling them with fluorescent dyes to provide high contrast for imaging in the clinic. In this slide here, we're showing a list of the different targets which we have developed peptides for, and we'll be using them in our demonstration. For colorectal cancer, there are two pathways. The alternate pathway is mediated by BRAF. The traditional pathway is mediated by mutations in APC, P53, and KRAS. And depending on which pathway, the cancer has either a predominant location in the proximal or right colon, or in the distal and left colon. And so this is something we need to be aware of when we do the procedure. This is a peptide developed with specific binding to peroxyredoxin 1. It's a 7 amino acid peptide that is labeled with FITC that provides fluorescence at around 510 nanometers in the visible regime, and this was chosen in order to be compatible with our imaging instruments. Here's an example of how we use this peptide in the clinic. In the left is a white light image of colonic mucosa. There is a serrated adenoma in the center of the image. However, if you compare the structural changes with that of a normal appearing colonic fold, it's quite hard to tell them apart. The difference is quite subtle. However, after we administer the peptide, and this is done topically, you can see bright contrast from the serrated adenoma in the center of the region, and there's really no signal from the mucosal fold. So this is an example of how fluorescently labeled peptides can be used to detect premalignant lesions in the colon. The instrument used in this case was a colonoscope developed by Olympus Medical Systems. So they were our industrial partner for this project, and they adapted the detection filters in order to highlight the emission spectrum from the FITC dye. Here's an example of how we can use confocal endomicroscopy. You can see the instrument coming out into the wide field image on the left and coming into contact with, this is a tubular adenoma. And once the instrument is in contact with the epithelial surface of the adenoma, you can see the optical section on the right in the confocal image, where on the left side you're seeing the premalignant crypts that are intensely stained by the peptide, whereas the normal tissues directly to the right is fairly absent of staining. The confocal endomicroscope is made by our industrial partner Mauna Kea Technologies, and this instrument passes through the instrument channel of the endoscope, where it can come into contact and collect images instantaneously that appear as histology. And we're using the peptide to stain the crypts to highlight which ones are actually dysplastic versus normal. Now let's move on to early detection of esophageal cancer. Like the colon, the esophagus also has cancer that arises from premalignant tissues that are flat in appearance. This occurs in the setting of a condition called Barrett's metaplasia, which is a transformation of the epithelium from squamous to columnar. It actually appears as a salmon pink color on white light endoscopy. However, the crypts can be dysplastic and lead to cancer. However, it's very hard to tell on the white light image. We have performed gene expression profiling to look at the potential targets for imaging. Esophageal cancer is a heterogeneous disease. It arises from genetic predisposition, but in addition from reflux of acid and bile. So we've identified EGFR and ERBB2 as two promising targets for early detection in esophageal cancer. Now, because of the increased heterogeneity, we have developed a multiplex detection strategy where we're using not one, but two peptides, one for EGFR and one for ERBB2. They are labeled with two different organic dyes, Cy5 and IR dye 800, which has minimal spectral overlap in the fluorescence emission band, and this allows us to distinguish the two targets from one another. Here's an example of how we use these peptides for clinical imaging. In the video on the left, we're looking at the white light images from the human esophagus, and then on the right, we're seeing the expression pattern for ERBB2 in the red and EGFR in the green. We also collect a reflectance image to provide the anatomy of the esophagus, and then the merged image is shown in the lower right. These images were collected with a scanning fiber endoscope that scans in a spiral pattern to create the image. It is also an accessory that passes through the instrument channel and is used to collect the fluorescence image. The instrument plugs into a base station that sits on a cart that wheels into the procedure room. We've also been able to collect confocal endomicroscopy images. As you are seeing here, the confocal instrument is coming into contact with the esophageal mucosa, and different expression patterns are shown for squamous, Barrett's, high-grade dysplasia, and esophageal adenocarcinoma. So what are the current challenges for fluorescence imaging for early cancer detection? One is that cancer is molecularly heterogeneous, and the general practice physician needs availability of both imaging agents as well as imaging instruments. What are some potential for future solutions? Well, we've already demonstrated the ability to multiplex detection of two targets and can extend that to 3 or more. And the solution is to partner with pharma as well as the medical device industry. I'd like to thank the National Cancer Institute for funding to support this work. In particular, I'd like to acknowledge the Barrett's Esophagus Translational Research Network, Consortium in Imaging and Biomarkers, Network for Translational Research, as well as the Academic Industrial Partnership. Thank you.